Okay, if everybody is ready, we'll get started. Actually, a few announcements before I introduce today's speaker. First is that this is, at least we believe, the last DCRI research conference for the summer. Typically, there's a block of time where people are on vacation and traveling and a variety of things. So it's time for us to regroup and to gather your suggestions for speakers, topics, programs, formats in the coming year, which typically would be after Labor Day. So that's the first thing I'll share with you. Second is that the DCRI and Duke overall is going to be changing its CME process. Instead of using a computer like we have in the back, when you come into conferences, there's going to be a code that is provided, and you will do it via text messaging through the CME office. But you have to know the code, which would be specific to a particular forum, like the DCRI Research Conference on June 25th would be a particular number. And in order to get the number, you have to come. There's no other way that you could get credit for it. So we'll let you know in advance when that's going to take place. But if we have special conferences during the summer, that is the system that we will be using. Third is that, as I mentioned last week, there are microphones in the ceilings now, so that we don't have to pass around a handheld microphone for questions and answers. We're working on the sensitivity a little bit, because as it turns out, when people are eating and opening up things, we pick it up, and on the archived version, it was all there. This has nothing to do with the NSA. So today's speaker is Soko Setoguchi. She's an MD PhD and also has a doctorate in philosophy. She's an associate professor of medicine here at Duke University Medical Center, spending most of her time at the DCRI. She's a pharmacoepidemiologist and cardiologist by training, which, from my perspective, is a perfect balance of a person being able to understand disease and its treatment. She also has appointments as an adjunct professor at Harvard and the University of Tokyo. Her major area of interest, which she's going to talk about today, is comparative effectiveness, utilizing her very interesting and what I think is a very important background. So thank you for joining us today.
Thank you very much. All right, so thank you for the opportunity. I'm going to start out my presentation with a quiz. So the question is, the best evidence come from which of the following types of studies? 1, systematic reviews and meta-analysis. 2, randomized controlled trial. 3, prospective cohort studies. 4, observational cohort or retrospective cohort studies using databases. 5, case control studies. 6, case reports. And 7, all of the above. 1, 2, 3, 4, 5. Some people counting best twice. Okay, 6, 7. Okay, I can probably stop my talk now and then take questions, because this is a message I want to sort of get across. So this is sort of a typical pyramid that you see, sort of rating types of studies in terms of validity of the evidence. And then it's been said that the systematic reviews and meta-analysis is the best evidence. Second is the randomized trials, and then followed by observational studies. And I would like to sort of toss out this idea. And then to me, that the form of clinical research providing best evidence depends on the nature of the questions, clinical or policy questions. And then also I'd like to use the quote from Sackett: the best evidence is usually found in clinically relevant research that has been conducted using sound methodology. So again, methodology, whatever that is, meta-analysis, randomized trials, or observational study, that's probably the key to get the best evidence. So my talk, this is an outline of my talk today. I'm going to talk a little bit about epi, pharmacoepi, and then go on to sort of give you some example about application of pharmacoepi method into CER compared to effectiveness research. And then I'll sort of chose 3 topics around here. I had more than 3 that I wanted to talk about, and hopefully I can cover all of this in my talk. And then at the end, I'm going to talk a little bit about the ongoing study that we have in my group for Global Research Network in Pharmacoepi. So what is EPI? Going through medical school, going through residency and fellowship in Japan, I never thought I would become an epidemiologist. Epidemiology was sort of an alien field to me as a clinical clinicians or medical students. So when I talk to people, I'm an epidemiologist. People often think I am a dermatologist, epiderm, right? Which is not the case, which is not epi. Or some people think that I'm like a cool woman chasing a mysterious infectious disease, because epi is known for infectious disease epi most of the time. Epi is the study of the distribution and the determinants of disease in human populations. So the key word is the disease here. Sometimes that relates to the treatments and beyond the diseases, but also the population is another keyword here. We always deal with a sort of large population in epidemiology. So EPI is not all about measuring incidence and prevalence of diseases, understanding disease risk factors, prevention. This is the key components of epidemiology. But to me, as a sort of researcher in the clinical research or pharmacoepi, EPI is really just a powerful tool to help us conduct sort of valid research. It provides a framework to understand or conduct valid observational research. So methodology is really the key component of epidemiology to me. When you see relative risk of greater than 1 or less than 1, or risk differences less than or greater than 0, it doesn't necessarily mean that that's the cause or treatment is effective or safe. So you first have to exclude a chance. Of course, you have to use knowledge in biostatistics to be able to understand what chance means and when you can exclude it as a chance. Also, the next step is to understand the bias, because observational study is inherent to biases. And then after you do that, you can finally say there might be a causation between exposure and outcome or treatment and outcome. In terms of bias, I think epidemiology provides a best framework in terms of systematically assessing the kind of bias and then how that matters in a clinical research. So just moving to the next topic, which is a little bit about pharmacoepi and why this sort of field of study came to do sort of more methodological research. So as you know, we assess efficacy and safety in a pre-market setting. There's phase 1 and 2, 3 trials before the drug gets marketed. But we also understand that randomized trials is not a great source for assessing safety of medications. There are several reasons to that. The first thing was the small sample size. So trials are very expensive. You have to maximize the resources to spend. So we tend to have a very small sample that enough to achieve the specified, effect size, to achieve the power for the specified effect size. However, this also means that you cannot really detect or rare safety events. Safety events tend to be rare, and then you might actually see 1 or 2 safety events in the trials. Then you would also have a very unstable estimate. You cannot really get out of anything. Another issue with a trial is a short follow-up. Again, we tend to sort of look at the surrogate outcomes and not real clinical events, and therefore the short-term follow-up is very short. And then if that's the case, in terms of looking at the safety endpoints, you really cannot detect delayed or long-term safety adverse outcomes. The next point is non-representative selected patients to minimize, again, maximize the resources, maximize the chance of detecting the efficacy, size effect. I think we tend to eliminate the noise by doing so, by selecting very sort of healthier population who has the disease but not other comorbidities. We tend to exclude elderlies. We don't include pregnant women. We don't include children. So because of that, those are the vulnerable population for the safety endpoints. What we see in the trials is typically underestimate of the real risk in general population. Finally, intention treat analysis is a sort of the standard method of analyzing the trial data. This is we say that this is a very conservative way of analyzing the data in terms of detecting efficacy, but it's actually not conservative in terms of detecting safety endpoint. It's conservative because you underestimate the efficacy, but if you underestimate the safety signals, that's not conservative. Thinking of safety, another thing to come to your mind, it would be an adverse event reporting system. Safety events ranges from the immediate and clinically easily recognizable to delayed, hard to recognize, recognizable events. So one typical event that is clinically easy to recognize is a digitalis toxicity. So this was found in the Foxglove plan. And then around the same time this treatment effect was observed, Dr. Weathering also observed that the toxicity, clinically observed the toxicity and described it in the literature in early 70s, I believe. Another typical safety endpoint is phacomelia. This is a German bass baritone singer. He's very good actually, but he was born with phacomelia because mother took thalidomide. And this thalidomide caused a lot of issues. You know that in the United States, I forgot her name now, the FDA officer actually prevented this drug to be marketed and preventing thousands of phacomelia cases in the United States. So these types of events can be easily captured in adverse reporting system because in the adverse reporting system, we rely on clinicians and patients to report the events. If clinically recognizable or not connected to the drug, it won't be reported in that system. So these types of event, because it's delayed, mother takes the drug for morning sickness, 9 months later, kids are born with phacomelia. And it's really hard to connect that event, drug exposure, and the outcome. And then that's why it took a while for us to understand the effect of this drug. So spontaneous reporting system is great because it's relatively quick, and then it will be, the data can be gathered soon after marketing. And then it's great to detect the clinically apparent adverse events with large effects. However, it has a limitation because it cannot give you the rate of the events. We only get case reports, but we don't know the base population. So you cannot really determine the rate of the events. Next, because of that, you cannot really determine the effect size, how bad the drug is in terms of causing some certain side effect. You never know from the adverse reporting system. Of course, detecting delayed or long-term effect for adverse events and small to moderate risk is really hard to do in this reporting system. And therefore, there's a need for formal safety studies using epidemiologic method in the post-marketing settings. So I think pharmacoepi field sort of evolved over the last 30 to 40 years. We relied on large databases to conduct post-marketing studies. In terms of ideal characteristics of data for post-marketing drug safety studies are here. So it has to be large because, as I said, safety events tend to be rare. It has to be population-based because vulnerable population, we want to include the vulnerable population such as elderly, people with comorbidities, and children and pregnant women. We want to have a very good data on drug use because that's the main exposure that we're looking at. And then nowadays, most of the drug for chronic use, not like antibiotics for short term, but many drugs are chronic use. So you really want to have a longitudinal information on drug use. Of course, we're looking at a safety events, so we want to have accurate data on adverse events. And then, of course, we're dealing with observational studies, so we're amenable to biases. So we want to have details and accurate data on potential confounders. And then because we were trying to find safety signals in a post-marketing setting, we really want to solve the issue if there's anything promptly. So we want to have data available promptly. And then in the field of pharmacoepi, we used large clinical administrative databases for the last 30 years or so in terms of conducting safety studies. The characteristics of that database is large, as you know, longitudinal. Data already exist. It could be retrospective. You can also expect that that data would accumulate over time, and then you can conduct prospective study using that data. We have good data on drugs, that we think. It's a detailed longitudinal pharmacy dispensing data. and then capture important clinical interactions so that it captures major clinical events such as cardiovascular events or other major deaths and so forth. A little bit about drug data. You might not agree when I said the administrative data has a good drug data. Thinking through what we really want to know in terms of exposure is if the patient took a pill or not, right? Not if the patient dispensed a drug or not. So that would be the best information we would get. Thinking through how patients would take the medications. So they have to go to a doctor to get prescriptions and then go to pharmacy to get the drug dispensed, open the bottle, and take the pill. So thinking through this process, we can see what the data we can get in terms of drug exposure. To know what we really want to know, I don't think there's any automated databases for that. You have to either directly observe or measure blood or urine level of the metabolites, or you can ask patients if they took it or not. Next level is opening the bottle. You can count the pills in the bottle, or you can put a small electronic device to monitor bottle opening, which is very expensive and it cannot be done in a large population. So then the next sort of surrogate for drug exposure would be a pharmacy dispensing data. And this is the data that you can get for millions of patients if you have access to the data. And then, of course, you can also get prescription data from EHR or other sources. But to me, that's sort of even farther away from what we want to know. So that's why I sort of say that the pharmacy dispensing data is probably the best source for drug exposure for large populations. So now we think that the claims data would meet the top three: large millions of people, population-based, and good data on drug use. Available promptly, it might depend on your situations and then data sources too. And then accurate data on adverse event, it depends on what you're looking at. You cannot identify everything in claims data, but certain events can be identified reliably in claims data. And the last one is, of course, no. We only have a claims diagnosis procedure code. We don't have lab data. We don't have values for lab. Also no signs and symptoms or anything included in claims data. So in terms of adjusting for confounding is the biggest weakness of the claims data. So that means that if we use claims data, we only get garbage out. So my answer to that is no data is perfect. Again, I think we've already used these data and demonstrated that we can produce useful evidence for certain safety events and certain questions over the last 30 years. I think it's obvious by looking at the literature. And I think pharmacoepi studies using large claims data enabled assessment of medication use and safety using formal epi studies with fast turnaround in population-based setting and vulnerable populations for rare events, long-term outcome with continuous monitoring. Now we're shifting towards more about the risk management paradigm in terms of drug safety out there, and then also focus on the life cycle, product life cycle, managing risk throughout product life cycle. Even with that, I think claims data have a very important role as shown in mini-sentinel efforts and other efforts as well. So now, the last one. So this no actually led to great interest in the field into methods, because we're dealing with imperfect data. We really want to understand the data and then want to be creative in terms of how we can get out of this data. And that's why we really were interested in methods, and then really came to understand the limitations of the data, challenges of database studies, and then improvement of the design and method to understand and control bias. And that sort of leads to my next talk about how these methods research conducted in the pharmacoepi field would apply to CER. Okay. So we've been talking about imperfect data, which is claims data now. We know that, and then we've been sort of aware of the fact that we've been trying to create a better data through data linkage. This is sort of the idea, schematic idea of that. This part is, so claims data typically consists from three data components: enrollment data, pharmacy claims data, physician and facility claims data. So this is a typically available component. Sometimes we can use this data as sufficient to answer certain questions. Sometimes this is not sufficient at all. So the idea is for each project, we have to assess the data components, what we need to be able to conduct valid studies. If not, we might want to bring in different data sources from other places to create a sort of better data for per project basis, So this is a very early study that I did. I was still a doctoral student when I started looking at this, probably like early 2000, 2003 or 4. We were interested in looking at the risk of statins on cancer. This question sort of emerged from the trial published around that time, Prosper, looking at pravastatin showing increased risk of cancer in trial data. And then soon we realized that the claims data is really not a good enough data because identifying cancer in claims data is really tricky. And then we decided to link Medicare data with pharmacy program to state cancer registries in 2 states, Pennsylvania and New Jersey. This time, there was already a linked database between cancer registries and Medicare data existed called CER Medicare data, but we couldn't use that data because this time Medicare data didn't cover drug benefit. So we had to link this special data we had from the 2 states to the state registry there. We used Social Security numbers to link the data. This provided not only the accurate identification of the outcome, but also the stage information for the outcome so that we could also look into the possibility of detection bias. When you find more sort of cases for drug users who are sort of early stage, you sort of suspect that the drug users might have more detection bias there. So those things are very useful in this study. And then this was sort of the entry to linkage world for me. And then around the same time here at DCRI, or a little bit later, while I was sort of struggling to link data without Social Security, the work was going on here, later published here. Leslie and Brad is sitting there too. But at the same time at Harvard, when I was still at Harvard, I was also trying to use a similar linkage methods to link cardiovascular registry to Medicare data. And later, I had a chance to validate this method through a HRQ-funded study. And then I linked, we got a registry directly from CMS, which included Social Security numbers. So we were able to test different linkage rules with and without Social Security numbers, and then test the validity of the linkage without Social Security, where we're only using gender, date of birth, admission date, and provider ID. And then comparing this linkage rule to gold standard, you'll be actually surprised because 100 means on all of this means perfect. And then, so this rule actually provided sensitivity of 95%, 98% specificity, and positive predictive value of 98%, almost close to perfect, compared to the gold standard linkage, which included Social Security numbers, provider ID, and admission date, which was the sort of minimum necessary components, linkage component to make each record unique because we're linking at the hospitalization level. So more work is actually going on here, I think by Sean O'Brien in outcome group. And then also we were also working on comparing probabilistic linkage to deterministic linkage, which is this linkage, versus sort of considering the uncertainty in terms of the accurateness of the linkage variables. Okay, next topic is bad information. So we all know that the bad information, inaccurate information in data would cause bias. But sort of we really want to know more than just that, because we want to sort of assess the impact of that. If it's minimum, who cares, right? So I think EPI sort of provide the basis of understanding the validity of the information and how much impact it has on the estimates of compared to the effectiveness studies. So to do that, you have to actually do some extra studies looking at validity of, let's say, claims-based diagnosis of stroke versus gold standard diagnosis of stroke. What we tend to do when we don't have access to do it in one study, because you have to have both information in one population, which sometimes very difficult to get. But we say we found X% of stroke patients in this database versus we found similar percentage of stroke patients in this database, let's say registry. Then we think that, okay, we may be actually capturing the right number of strokes, and then therefore the diagnosis of stroke in claims data is right. That's actually not enough. Finding the same similar size in both databases would help, but what we really want to know is the size and the overlap. And once you have that information in one population, you can calculate all sorts of metrics like false positives, true positives, false negatives, true negatives. Why this is important? Because these actually let us to calculate sensitivity, specificity, PPV, negative predictive values, right, of that specific diagnosis or definition of diseases. And then that actually can be used to estimate how much bias you would get in your estimate of safety or effectiveness, comparative effectiveness. So this is a study I did back in 2000, publishing back in 2008. I was able to get this estimate from the validation study, and I was able to apply this to my actual study to understand how much bias I would have using this definition with this sensitivity and with this specificity. Another example that I want to show you was a study that we conducted under CV Consortium here, trying to validate stroke diagnosis in Medicare patients. So last time the stroke was validated in Medicare was using the data before 2000. So it was quite old. So we wanted to sort of see if this, what they found, which was a very accurate, high accuracy of stroke identification in terms of specificity, but sensitivity was a little bit lower. We wanted to see if that still holds in the contemporary Medicare population. So we collaborated with University of Alabama, Birmingham, who was funded for what's called REGARD study. I forgot what acronym stands for, but this is a study to this is a research funded by NIH to study stroke risk factors in general population. They oversampled Black high-risk patients. So they had 30,000 patients in REGARD study. We tried to link it to Medicare using Social Security numbers here. We had to go through the consent process, so we lost some here. And then at the end, we were able to link half of that patients to Medicare. And using this population, we validated stroke, comparing claims identified stroke to stroke identified through adjudication process in REGARD study. So this is a little bit of a complicated slide, but notice that what we have here in terms of all the sort of population, we had 15,000 patients. We categorized them into four levels, four categories. One is the patient who had a claims identified stroke, and then also self-reported identified stroke, or just a self-report for stroke. And then we also had a patient who only had self-report but no claims. And then also there's a group of patients who had no self-report but claims only for strokes. And then, of course, before REGARD started adjudicating stroke, before they linked the data to Medicare. So what they actually adjudicated was just the people who had self-report. So in this category, the second row, wasn't something that they validated before. So if we assume that the people didn't have stroke, no stroke, then these 97 patients didn't have stroke. So what we did under CV Consortium is to sort of sample these patients to validate, adjudicate stroke, went back to medical records and adjudicate their stroke status. So what we found through that is that this is very understandable. People who had claims and self-report, 90% of the patient had stroke. And then also people who had self-report only, majority were not stroke, 90% were not strokes. But surprisingly, people who didn't have self-report but identified by claims only, more than 90% had stroke. So that means that if we were actually just relying on self-report, REGARD study was missing some true stroke cases. So considering all those information we had, we tried to validate. And then also another implication of this small sample that we did was if we didn't consider this group, that we would erroneously concluded that Medicare claims identified stroke had a lower validity. So considering all of this information, that we found that Medicare identified strokes with a very high specificity and high PPV, and as we knew that the sensitivity was a little bit low, like 60%. And then just flipping this side, our goal was to validate the Medicare identified stroke, but thinking about self-report triggered stroke identification in REGARDs, I think it's perfect, but also, because of the nature of how we sort of sampled and what's validated, but it also turned out that they are not perfect in terms of capturing all strokes in REGARDs data, 90%. So again, no data is perfect. The last topic in the method is confounding by indication, healthy user bias. I have to look at what time it is now. Okay. And then I guess I have so many topics that I want to talk about and then in this field, because this is big thing in pharmacoepi. As I showed you, claims data don't have good information in controlling confounding, and this is the thing that we struggle most in this field. So a lot of research is going on in this field, and a little bit about confounding, as you know, that it's a factor that is associated with treatment or exposure and outcomes that are not on the intermediate factor, on the intermediate path to the causal pathway between treatment and outcome. That would distort the association between treatment and outcome if not considered for controlling or adjustment. So propensity score is one of the very popular methods to control for confounding. And then a lot of research actually happened over the last 10 years in terms of refining propensity score methodologies. And then I know that our group also have done a lot of work there. In terms of propensity score, it's a two-step methodology. First, you have to estimate propensity score for treatment, and then you would have to consider those propensity score in the outcome model. Objective is, again, control for confounding. Propensity score is a predicted value for treatment for each individual, and it's a two-step approach. First is to develop PS model to estimate propensity score, and then the second step is to use PS for outcome estimation. So typically we use logistic regression for PS model. What difference between the multivariate modeling we usually do is that all the confounders goes to this first model, not the second outcome model. And then you would estimate propensity score for each individual. And then once you have that, you have few options to control for confounding. You can put that in an outcome model like that as a continuous propensity score or deciles or quintiles. You can do stratified analysis, or you can do matching. In terms of developing propensity score, you sometimes wonder if we should include interaction terms here or what sort of model we should use, that kind of thing. And then one of the things that I did early while I was sort of working on the propensity score model was that to assess the relationship with C statistics of the propensity score model and then bias control. And then because at that time people were saying we have to get the C statistics for propensity score as high as you can, and then you would have a very good propensity score model. And it wasn't actually clear to everybody if that's the case, but people sort of started doing so. So I did a simulation study trying to look at develop a model with different C statistics using data mining strategies like neural networks, recursive partitioning, or logistic regression model with lots of interaction terms, and then found that there's no association between the amount of bias you can control and the C statistics here. However, there was actually a clear association between the amount of standard errors that you get in the estimate versus C statistics. So it's not necessary to have a very high C statistics for propensity score in this case. Another thing that I worked with a student at Harvard was adjustment. So one of the advantages of propensity score that you can put lots of variables into the model, whereas you have sort of a limited ability to do so in multivariate outcome models. But by doing so, are we hurting ourselves by inducing different types of bias, which called M bias? Just very quickly, so this is what we call causal diagram, like similar to what we saw the triangle before for confounding. When you are interested in assessing the exposure and outcome relationship, and if you want to include, so in this case was SSRIs and lung cancer, if you want to include coronary artery disease in the adjustment, which is a factor that has a direct association between exposure or outcome, but through a different factors like depression and smoking like that, which sort of, that's why M come from, this sort of structure of the causal diagram is M, and then when you adjust for such factor in this M structure, you would actually, theoretically, it's known to cause some bias in the outcome model. So what we did is, again, simulation studies looking at how much sort of effect we have to have between these variables for us to have a very significant amount of bias here. And then it turned out that most of the time when you see association, which usually you see like risk ratio of 2 or 3, it doesn't actually matter too much. You only get very small amount of bias here. However, when you start seeing a very large effect here, like 27, 15, something like that, then you would get like 20, 30% of bias in your estimate. So bottom line here is I think this bias is theoretical most of the time, and you don't have to worry too much about it when you're developing propensity score. So just quickly, using probably the rest of the 10 minutes, I'd like to sort of shift towards more about the project that we're having ongoing in our group in terms of connecting databases globally. So this is actually my thesis, which I looked at the safety of TNF agents and on cancer outcomes. So for those who work in this area, you know that the population size shrinks really quickly. You have a very large databases to start from, and then you start looking at a certain population, and then you start looking at a certain population with certain exposure, and then you look at a very rare outcome. The size really shrink very quickly. And then there's a need to get more databases or more numbers there. And that's where we sort of thought about pulling sort of multiple databases from 1 to states. And then we didn't have enough numbers here. You look at the number, biologic exposure is only 400 here. So we tried to pull the data, non-US data, Canadian data, British Columbia, and then we got this number. Pulling these databases also sort of make me think about the variability of the results that you get from different regions. So looking at this is one database, 2, 3, 4. The SAD database, we sort of, because Pennsylvania and New Jersey only included patients over age 65, we sort of divided the population into 2 here for British Columbia. But look at these estimates for different cancer outcomes that really varies a lot. I mean, partly because of the rare events and then wide confidence limits, but it would make me think that other factors we have to consider in terms of analyzing the data from multiple regions. So this is sort of the entry to multi-database study, which I did personally in a small scale, but I know that there's a lot more going on out there, has been already going on, and the most famous one might be HMO Research Network. Now it sort of became the basis of Mini-Sentinel. OMAP is another one, sort of Canada has multiple one, and also Europe has multiple research networks. So what's going on here is we actually got funding to sort of develop infrastructure and do some very descriptive research in using Asian databases. It's an industry-funded study, and then this sort of relates to the, I guess, effort that I've been doing within the International Society of Pharmacoepidemiology and working with multiple collaborators in Asian countries to develop this network. So why Asian database network? Because I'm Asian, first of all. But this is where I was actually back in late 90s. I did 1-year internship in general medicine, had a opportunity to sort of observe practice in Japan versus US. And then what I noticed is the dose of medications is very different. So in Japan, sort of basic study, over-the-counter medication, acetaminophen, we use up to 1,500 per day, whereas you use 1,000 milligram every 6 to 8 hours. So it's like twice or 3 times more. Heparin and warfarin as another drug, I was sort of stunned to see the differences. Recommendations are similar in both countries, but in terms of sort of the dosage that the patients use, Japanese patients achieve the same therapeutic target with maybe a half or 1/3 or 2/3 of the dose in American patients. So variation exists here. So this is a more formal research published in American Journal of Cardiology that the Japanese, this I think data from trials, and then show that the dosage is half in Japan versus United States for most statins. And then they achieve the same therapeutic benefits with lower dose. Another reason why Asia, because we have medications that are not available here or uniquely used in Asia. So one of that is a Chinese medicine in Japan that the use has been increasing over the time. It's a prescribed medication covered by health insurance. I can prescribe Chinese medicine even though I didn't learn much about Chinese medicine in medical school, which is scary. And then, so there are racial and ethnic influences on pharmacokinetics and pharmacodynamics, and then clinical practice is different. There's a different dosage recommendation, different indications, compliance, off-label use. Also, policies affecting physicians' behavior in terms of prescribing would be different in multiple regions. Also, another reason is, as I said, some drugs are only marketed in Asia. Chinese medicine is an extreme example. There's also NSAIDs that are not available in the United States, but commonly used only in Korea and Japan as well. So those things are sort of speaks to the need of own safety and effectiveness data in Asia. So we have a sort of group of people, researchers within the International Society of Pharmacoepi, formed a group called Asian Pharmacoepi Network, and I'm one of them. So the goal of this network is a voluntary effort by research from multiple countries, has been going on over the last 4 or 5 years, and it's developed a system to assess safety and effectiveness of medications, vaccines, and medical devices in post-marketing settings. So really, our goal is to understand the role of race, ethnicity, culture, also have our own safety evidence. This is actually a scheme for Mini-Sentinel. Now you can see a very good structure here that the FDA is funding, giving the money. There's a coordinating center with multiple people, and there's multiple data partners. A lot of money flowing, and then they have a very good infrastructure. Lee's contributing a lot in here. What we had in Aspen, as I said, it was a voluntary effort. We had 2 people in coordinating group, and then we had maybe 2 or 3 people in each sort of database. So there's one database in Japan, another one in Taiwan, Korea. We also had Australia, and then Sweden, and then US. So this has been going on over the last 5 years, but we've been really suffering from lack of funding and then sort of secure time to work on this. And now we have opportunity to expand this work. So we have an industry funding here at DCRI, to help develop infrastructure, like a common data model and mapping codes suitable for multi-country research, and then conduct sort of broad descriptive analysis to gain insights of health status and health service use in East Asian countries comparing to US. And then we have a voluntary participation from 4 Aspen sites, Asian Pharmacoepi Network sites, Japan, Taiwan, Hong Kong, and China. So this actually just kicked off over the last few months, and then we're very excited about this opportunity here. And then really understanding that methodologically, what's most interesting will be to understand the source of heterogeneity and then how to deal with it. And this is a recent article from the funding people, actually Patrick Ryan here, Paul Stang, and Jesse Burley, and J&J. They are the part of the collaborator, so, and then funding people, and we're very excited to work with them. So at the end, so what? I hope I had a sort of good story convincing you about the relevance and usefulness of pharmacoepi method in CER and clinical research. There are opportunities for global safety research and effectiveness research as well. And last thing I would like to do is selling the field of pharmacoepi and inviting to annual meetings. We have annual meeting for Society of Pharmacoepi, especially to the meeting in 2014. Why? First of all, this is a very field that is sort of growing interest. We started out in 1999 with 400 participants in the annual meetings, and now we're up to 1,200. I think this year, it's happening in August usually, and then we alternate North America and Europe in terms of the conference sites. And then our really focus is safety, risk management, and then methodologies as well. So I know a couple of clinical researchers who actually come to these meetings to sort of get exposed to methodology, more recent methodologies, and then educated for methodologies. Why 2014? It's the 30th anniversary of annual meetings, and ISPI is going to Asia for the main society-wide conference first time. And they actually been recognizing the importance of the Asia-Pacific region in the field. They created a chapter, and then they added a board member from Asia, so on and so forth. So they finally decided to go to Asia for the main meeting. And then another thing is I'm actually chairing the meeting, and then I'll be responsible for scientific programs. And I like a lot of sort of participation from Duke, and then sort of emphasize the work that we do here. So this is sort of the advertisement for the 2014 meeting. As I said, the meeting usually happens in August, but it'll be in October this time in Asia because August is a typhoon season. So that's all I have.
starving for the right data. Can't we come up with some type of national or international resource instead of trying to mix and match and taking a little bit from Medicare claims and a little bit from action and a variety of things that people do? They're hybrids. Is an electronic medical record one way to go about it, or does it require literally an act of Congress in the United States to make sure that it has very important information?
Yeah, I think the latter is true, the answer. Part of the problem we have in the United States is that we don't have even claims data covering the whole population. It's fragmented here and there. In terms of going to EHR, so far I think it's still under development. It'll probably take another X number of years for that to be useful. I had some experience using EHR data, which hasn't been very pleasant and useful so far.
Yeah, great talk, Soko. So Aspen, as your scan, as you described it, it sounds like it will be an opportunity for OMOP and other investigators to ask questions. Do you expect that there would be opportunities for DCRI fellows, investigators to pose questions, or is that in the future?
I think so. So right now we're in the phase of developing infrastructure, and then before we have infrastructure, we really cannot really do studies in terms of answering clinical policy questions. But once that happens, which is going to be after a year or so, I think in the second year we probably have opportunity to do so now. Yes, if you're interested, please contact us.
One of the cardiology fellows, I had a question. So yesterday I saw a patient who refused to take aspirin a day, even though he had bad CAD, but continued to smoke and ride his motorbike. So there was he had difficulty perceiving risk, which a lot of human beings do. So especially in pharmacoepi, where if you label a drug having a bad effect, even though the effect size is extremely small, public perception is generally pretty either too much or too little. So how do you control for the message that gets out of your findings?
That's a very good question, and I think we sort of, in the society, we started sort of really approaching that question with more about risk communication, also risk-benefit profile of the medications. So that is a topic, an emerging topic in the societies. I know that Europe actually has an advanced perspective on the risk-benefit assessment, and I'm not an expert in this area, but this is something that would evolve over the next several years, I believe.
If there are no other questions, I'll thank Dr. Setoguchi for a wonderful presentation.
Thank you.
Thank you for your